structure. As you know, in ear we have the external, middle, and internal ear. And the internal ear, also known as the vestibulocochlear system, the system which related with the hearing of voice and also related with the understanding the position of head. Okay, here you can see a section from head. This part is our auricle, continue with the external ear or external acoustic meatus. Here we have the tympanic membrane, and the tympanic membrane is the border between the external and middle ear. In middle side here we have a middle ear cavity with three ossicles, the malleus, incus, and stapes bones, and also the middle ear cavity continue with the auditory tube in the inner side. You know the origin of the external and internal acoustic meatus from our lecture related with the pharyngeal arcus development. And in outer ear or external ear, we have only the auricle and this passage. This passage surrounded with the epithelium, like our skin epithelium, stratified squamous and keratinized epithelium. Under the epithelium, we have connective tissue. And under the connective tissue, in outermost side of the external ear, we have the elastic cartilage. And in inner side, this skin connected with the bone. Here you can see the bony part of the external ear. Here you can see the structure of tympanic membrane. It has two surfaces. The external surface covered with stratified squamous epithelium, and also it has internal surface which covered with simple cuboidal epithelium, and between these two epithelium we have connective tissue composed from the collagen and reticular fibers. In middle ear we have these three ossicles. The malleus connected with the tympanic membrane and connected from other head with the incus and continue with the stapes. And the voices from this tympanic membrane changed into the mechanical force and continue from the bones to the vestibular window or oval window in this part. Here is the beginning of the internal ear. And also the middle ear continue with the auditory tube and connected with the nasopharynx. Here again we have the epithelium. Here we have the simple cuboidal epithelium in the beginning, and in the other side the epithelium of auditory tube changed into the respiratory epithelium, and under the epithelium we have bony and in some parts elastic cartilage structure. And here you can see the internal ear. This red structure known as the bony labyrinth. Bony labyrinth. In the inner side of this bony labyrinth, we have another labyrinth known as the membranous labyrinth. Okay? Bony labyrinth in the outer side, membranous labyrinth in the inner side of the bony labyrinth. And between the bony and membranous labyrinth, we have a cavity. In this cavity, we have a fluid. This fluid known as the perilymph. Perilymph present between the bony and membranous labyrinth. The bony labyrinth composed from the bone and also divided in two parts, the vestibular part and cochlear part. In the vestibular part, we have the vestibule and also semicircular channels, present in this part. And also we have a nerve which is known as the vestibulocochlear nerve, which divides in two parts, the vestibular part and cochlear part can innervate in the inner side of the cochlea and vestibule. You can read these parts for yourself. Okay. This is our bony labyrinth, this yellowish structure. Here is our cochlea, bony cochlea or bony labyrinth of cochlea, and this part is our vestibule. And here you can see the semicircular channels. We talked about this yellow structure. When we cut this part and take this bony structure away, we can see the inner side. In the inner side, we have a cavity. This cavity filled with perilymph. And also you can see this pink structure. This pink structure is the membranous labyrinth. Membranous labyrinth continue into the cochlea and form this helical structure. And also continue to the vestibular and semicircular side. And in the vestibular side, form the sulcus and utricum. Sulcus and utricum. And the utricum continue with these structures, semicircular ducts. Semicircular ducts, not semicircular channels. Semicircular channels related with the bony labyrinth. The membranous labyrinth have semicircular ducts, not channels. Okay? Between the semicircular ducts and channels, again we have perilymph. In the inner side of this membranous labyrinth, we have endolymph. Endolymph present in the inner side of the membranous labyrinth. In membranous labyrinth, in semicircular side, we have the ampullas, these swallowed areas present in the membranous structure, known as the ampullas. And also you can see the semicircular ducts. In the inner side of ampullas, we have structure related with the sensing of the position of head. And also in the inner side of the utricle and sulcus, again we have a little same structure related with the position of head and head movement. And in the inner side of the membranous labyrinth, in the cochlear side, we have corti organ related with the sense of voices. Okay? Can you understand this one? Now we are going to talk about these structures and also the cochlear structure. When we cut our semicircular ducts and channels from this part, and look at the inner side of this structure, we can see the bony labyrinth in the outer side, the membranous labyrinth or semicircular duct in this part with endolymph in the inner side of it, and here you can see this cavity filled with perilymph. From one side, the membranous labyrinth should connect it with the bony labyrinth. Here you can see that side. The membranous labyrinth connected directly with bony labyrinth. When we cut the cornea and look at it in this part, again we can see the bony labyrinth. In the inner side of bony labyrinth, we can see this structure, this pink area. This area is our membranous labyrinth. The bony and membranous labyrinth form this structure in the inner side of the cornea. Three different cavities. Here is our cochlear cavity or middle cavity filled with endolymph, but these two cavities filled with perilymph. Okay, perilymph. We will talk about these cavities and structures. In vestibule, we have utricle and succor. In utricle and succor, we have a sense structure. The name of sense structure or sensory structure in the utricle and succor is macula. In semicircular channels, we have semicircular ducts, and in the ampulla side of semicircular ducts, we have crista ampullaris, spatial structure related with semicircular ducts. And in the inner side of cochlea, we have cochlear duct, this part. This is our cochlear duct. All of this structure is cochlear, but here is cochlear duct. And in cochlear duct, we have the spiral organ or corti organ. Perilymph, endolymph, we talked about that. Now we are going to talk about the utricle and succor, the structure related with vestibule. We have macula in this part and this part. Macula have a structure like this. In the macula, we have special cells known as the hair cells. Type 1 and type 2 hair cells present in the structure of macula and also cupula and also present in the structure of corti organ. Hair cells have special sterocilias and only one kinocilium, which have a role in understanding the position of the head. These sterociliums and kinociliums covered with special structure known as the otolytic membrane in macula. In the inner side of otolytic membrane, we have otoliths or ear sands and also gelatin layer. Here you can see. With displacement of these otoliths and gelatin layer, this special structure in hair cells can be active and transfer information via these nerve fibers to the central nervous system, information related with the position of head. And in the outer side of these hair cells, we have supportive cells. Here you can see these supportive cells. And these supportive cells have a role in supporting of the hair cells. All of these hair cells and supporting cells form sensory epithelium in the surface of the macula and also cupula. In semicircular ducts, again we have special structure with again sensory hair cells and supporting cells in this part. which form the sensory epithelium. But what is the differences between the cupula and macula? In cupula, we have endolymphic, we have a gelatinous structure without otoliths or ear sands. We have only this structure known as the cupula, and displacement of this cupula can active or inactive these hair cells and make some impulse to send to the central nervous system. This side of cupula, known as the crista ambularis. Here we have the connective tissue and nerve fibers connected with the sensory epithelium. Do you have questions about the cupula and macula? Can you understand the differences between the cupula and macula? What is the differences? Otolithic sand or otoliths or ear sands not present in the crista ambularis and cupula. Okay. Now we're going to talk about the most important side of the ear related with the hearing. Here you can see a section of cochlea. When we cut the cochlea, you can see different segments of cochlea. Firstly, you should know all of these ducts continue with each other. We have only one continuity cochlear system. But in sectioning, when we cut in transverse sections, you can see more than one cochlear system. When we look at only one bony labyrinth, you can see the bony labyrinth covers all the structure from outer side, and in the middle side we have the membranous labyrinth. This membranous labyrinth, also known as the cochlear duct or scala media. In upper side of the cochlear duct or scala media, we have scala vestibuli, and in the lower part we have scala tympani. Scala vestibuli and scala tympani covered with bony lamina and filled with perilymph, but scala media filled with endolymph. Scala media have a triangular structure like this. This part is the inner side of the scala media, and here is the outer side. In outer side, we have the ligament in this part, and this part is the stria vascularis. I don't talk about the role of stria vascularis and other things because you don't need that details. And this part is our Corti organ of Corti, related with sensing the voice. Which cells present in the structure of Corti organ is very important. You should know all of these cells, the location of these cells, and the number of these cells. When we look at this structure in higher magnification, we can see this part in higher magnification here. Here is inner side, this part is outer side. From inner to outer, we have different cells and structures. Firstly, you should know, this is our basilar membrane, this blue structure, basilar membrane, which separate the scala tympani from scala media. And also, we have vestibular membrane here, which separate the scala vestibuli from scala media. In after side, again, you can see the stria vascularis here and ligaments in this one. From inner to the outer side, we have the inner limiting cells here. These cells also known as the interdental cells. What is the role of these cells? Secreting of tectorial membrane. Tectorial membrane secreted with these interdental cells. Here is our inner sulcus, and in this part we have an outer sulcus. From inner sulcus, here we have the inner hair cells. Only one layer of inner hair cells present in the inner side, connected with the nerve fibers and support with pillar cells. In this part we have pillar cells which support these inner cells, inner pillar cells. Sorry, inner phalangeal cells, not pillar. Or inner supportive cells, not pillar cells, but inner supportive cells. A little in either side, here and here, we have pillar cells. Look at here, pillar cell. This cell and these cells. Inner pillar cell, outer pillar cell. These pillar cells form a tunnel, inner tunnel, between the inner side of the corti organ and outer side of the corti organ. This inner tunnel again filled with endolymph. In outer side we have the outer hair cells with 3 rows. 1, 2, 3 rows in outer side or outer hair cells. These cells supported with these phalangeal cells. Look at here, these cells is our phalangeal cells present in the lower side of the hair cells, in outer side. And these supportive cells extend their cytoplasmic portion to the upper side, to this part. And in this part form a shell to protect this inner side. And in this part, you can see a cavity between the cytoplasmic portion of the outer pillar cell, outer phalangeal cells, and outer hair cells. This cavity known as the Noel cavity. Noel cavity. A little in outer side, here we have supporting cells or Hansen cells. Supportive cells or Hansen cells present in the outer side of the corti organ. Corti organ composed of these cells only. Hansen cells is the last part of the corti organ. Again, here we have other supportive cells. The name of these cells in some areas is Claudius, and in some area known as the Butcher cells. And you don't need to name out that cells because this book or this reference book now talk about that structures. Do you have question related with corti organ? Can you understand the location of cells? Which cells have a role in the sensing? Hair cells, only hair cells, inner and outer hair cells. And these cells is our supportive cells or phalangeal cells, which cells form the inner tunnel, the pillar cells, inner and outer pillar cells, and which cells secrete the tectorial membrane, the interdental cells. Do you have questions? Okay. Let me look at the histologic section. What is the name of this part? Cochlear duct or scala media. What is the name of this part? It's not right. Vestibular, scala vestibuli. And this is our vestibular membrane. Okay? What is the name of this cavity? Scala tympani. And this is our basilar membrane or basilar membrane. What is the name of these cells? Interdental cells. What is the name of this structure? Tectorial membranes don't have any cell in this structure, have only the proteins and carbohydrate and fibers. And what is the name of this part? Inner tunnel. What is the name of this tunnel? Noel tunnel. What is the name of this cell? Inner hair cells. What is the name of these cells, these lung cells? Inner and outer pillar cells. What is the name of this cell? This and this? Outer hair cells. The name of these cells? Outer supportive cells or phalangeal cells. Okay? Can you understand? What is the name of these lung cells? The outer limiting cells or outer Hansen cells. Do you have questions? It's very easy. Easier than retina, I think. Here again, you can see same structures, scala vestibulis, scala tympani, the ganglion structure, the ganglion present in this part. And here you can see the portiorgan, and here is our scala media, vestibular membrane, and other structure. Here is our stria vascularis, only this one. Thank you for attention.